Hello, everyone. I'm your host, Ritvik Raya, and today I'm thrilled to speak with Dr. Jillian Silbert Paull, a PhD candidate in the Systems, Synthetic, and Quantitative Biology program at Harvard, and a researcher at the Sabeti Lab at the Broad Institute. Her work focuses on viral genomics from sequencing pathogens to improving global surveillance systems. Dr. Paull, thank you so much for joining us today.
Thank you for having me.
So, to begin, could you walk us through your path into biology? Like, what experiences early on led you towards studying viruses and genomics?
Okay, so I actually thought that I was going to be a mathematician for most of my life. I was obsessed with math as a kid. My parents joke I still today don't have much patience for art museums. So I kind of walk into an art museum. I quickly look at each painting and say, all right, cool, I saw it all. And then as a kid, I would sit down on a chair and just write out math problems to myself. So I had this, healthy obsession, I would say, with math. I went to summer camp for math, had fun with that, did a lot of math competitions. But I actually ran out of math right after my ninth grade of high school. So I did calculus my ninth grade and then, didn't really get to kind of continue more formally taking, I really probably should have taken college math and just it wasn't an option for me or something I thought about as an option. And so by the time I got to college, I was in this interesting place where I still liked math, um, but I also had a feeling that I might not, I'd been out of it for 3 years now and kind of wanted to explore. And so I took a freshman course at my undergrad university, which is Princeton, called the Integrated Science Curriculum. And it's this very cool course where it combines physics and biology and chemistry, math, computer science, like everything is all
Everything.
together. And so my goal was like, okay, I took math courses on the side. I still was like, I want to technically be able to go down the math path if I want to do that as my undergraduate degree. But I wanted to take this course also and see like what spoke to me most from the sciences. And very quickly, I found myself really gravitating towards computational biology. I think what I liked about math was that it kind of felt very practical to me. So I actually didn't like taking a lot of college math courses where it got very theoretical. I liked things having more of a strong application. And most of what you study in biology, like you can really see the application, right? Like we're studying human health, like we're trying to help people. Some biological research is more theoretical, but any biologist can take whatever niche thing they're doing and they can explain to you like how it impacts someone and how could one day, whether that's 1 year from now, 5 years from now, or 50 years from now, change someone's like the outcome of their life and like their health. So for me, really quickly, I kind of took this math part and was like, oh, I want to, see the mathy side of biology. So my undergrad major was molecular biology with a minor in quantitative and computational biology. And then when I was approaching graduate school, I at the time we call it wet lab versus dry lab. So wet lab is when you're still like in the lab doing experiments. Dry lab is when you're fully computational. I was still a mix, and so I wanted to be working with either viruses or bacteria just because they really fascinated me, and I enjoyed working with them in the lab. They're very easy systems to work with. They're very like, you can freeze them, you can take a break for the weekend, come back after the weekend, continue with your experiment. Like it's just very easy to run experiments with viruses and bacteria, and also again like that clear connection to human health really interested me. So when I was looking, when you start grad school, you typically, a lot of programs have you do rotations where you try out different labs, different projects, and all of my rotations had something to do with bacteria or viruses. And this lab with Pardees Sabeti was one of my rotations. I started graduate school within the first year of the COVID pandemic.
Um,
So, right, it was kind of a natural fit for me of, oh, if I anyways wanted to study viruses or bacteria, why not study the virus of the moment? And so I joined this lab and started doing a lot of COVID research. So that's how I got from math to viruses, and then genomic research is all through this lab here.
yeah. I definitely see the way you see it. Like, when I was younger, my dream was also to be a mathematician. Like, I wanted
Yeah.
to win the Fields Medal. And then
Wow.
right now I'm taking AP Pre-Calc, and I
hmm
just feel like the theoretical side of it, like, I don't know when I'll ever need like imaginary numbers in real life.
Yeah, it is funny because like as you do get to more advanced math, you suddenly realize like imaginary numbers does have practical applications. I can't tell you where because I don't remember anymore, but like my moment with that was it was linear algebra where I took linear
Yeah.
algebra as my first like freshman course in undergrad and I was like, what? I'm looking at these matrices of all these numbers, you're doing weird things to the matrices. I'm like, how does this have any impact? And then now, that you learn something in linear algebra called principal component analysis, and that actually can be used in almost like any other area of research because it just helps you take something that has way too many variables and like condense it into two important variables. And so it is funny where like
Yeah.
even those things that start to feel less practical do somehow connect back. But yeah, I think similar for me with you, like I just once I couldn't see it and visualize it anymore, I didn't like it.
Yeah. So you also mentioned how you worked in the Sabeti Lab on the coronavirus. So it
hmm
is known for its work in infectious disease genomics. Like, what drew you to this lab in particular, and like, what makes its approach like unique?
Okay, so I'll start with what drew me to the lab, and then hopefully I'll remember to get to what makes our approach unique. But for me, what drew me to the lab, so again, I joined during COVID, and the project I joined onto was a collaboration with a university in Colorado, where we as the lab helped them set up their entire system for how they were maintaining safety during the pandemic. And so I was literally getting like, samples that were from students who gotten sick on campus with COVID. I was getting their samples like that same week, sequencing them. We were analyzing the sequences, and then we were able to go back to the university and say, hey, here's what's happening on your campus. And so when I joined the lab, it kind of, like I said, like we have these rotations. It's like a probationary period where you're trying out the lab, and they're also trying out you and seeing if they like you. So I was only in the lab for a quick 10-week period. And in those 10 weeks, I learned an entirely new laboratory protocol and was suddenly left on my own sequencing all these samples, was on the computational side again, doing totally new techniques. And then also, maybe halfway through this rotation, we found basically that on this campus there was a variant of concern that was circulating. And so we reach out to the administrators and say, hey, we saw this thing on your campus. Just so you know, this has been flagged. I think it was in the state of California as like a variant of concern. It seems to have some properties that, health experts are concerned about. We want you to know it's on your campus. Like, let's just be a little bit cautious right now. Like, let's see what happens. And then we were able to tell them a few weeks later, oh, okay, we don't see this variant anymore. Your policies are working. You got this in place. Like it didn't spread more than we would have, it didn't spread so much that we'd be like very scared about this variant on their campus. And so for me to be part of that and like, 10 weeks is less than 3 months.
Less
So
than
in less than 3 months, I myself am learning all of these new protocols and skills and am also doing something that's meaningful for a community that's actually working, trying to move past this virus and trying to like have life back to normal as much as possible. So for me, like I just found that so meaningful. It's to see like what we're doing really matters. And it also matters beyond COVID, right? It's not just anything that we learned from COVID, we can now apply to other viruses as well. The second question was what makes our laboratory's approach unique?
Yep.
And what I would say is I think for me, what my perspective, what makes our approach unique is that we're really trying to come at this from every angle possible and to pull everything together. So it's very free, easy to have silos where, the doctors are doing one thing, people in their home are doing something else, the scientists who are sequencing the genomes, that's something else, and we're not talking to each other, we're not communicating about what's going on. So just to use this first project again as an example, we were combining everything. So we had a platform through cell phones, cell phone app for students to communicate to the administration of their school, but also to us as the researchers. What are their symptoms? What's going on? Like, how are they feeling? Then we're seeing their diagnostic results. So again, that's something that might have stayed in the clinic. Now, symptoms is correlate. We're connecting that in with the diagnostic results. Now we're sequencing their samples. We're connecting in the sequencing. We're also doing wastewater surveillance, looking for COVID through different wastewater spots on their campus, which dorms are having peaks in COVID per the wastewater. That's connected in as well. And so almost all of our projects, like if you look through like the papers, and like you have to like we have a lot of papers. We have a lot of different projects that we're in, but in general, we try to kind of combine as many different things together as possible, with the goal being how can we leverage all of this data to help people as quickly as possible and to slow down outbreaks. COVID in some ways is a bad example of that because obviously COVID is still here. But in the past, for most other outbreaks, like it really is something that like, if there's a measles outbreak, you want to contain that as much as possible. And we usually have seen in the past, like we're able to contain these outbreaks if we're able to understand them quicker and then implement policies that are appropriate, right? So not
Definitely.
every outbreak needs the entire world to shut down. So how what when do you need that and how do you figure out what you need, right? It's by quickly analyzing all this data and figuring
Definitely.
out how to leverage it.
So we're definitely going to talk about wastewater surveillance later, but for listeners who may not be familiar with viral genomics, could you give like a broad and like overview of the research you focus on?
Yeah, so the purpose of viral genomics, there's a couple different purposes. So first, there's I'd say maybe three main categories that you care about when you're doing viral genomics. One is, let's say someone's sick and you have no idea what they're sick with. How do you figure out what that is? So when COVID first hit in China, at first no one had any idea what this disease was. How do you figure out what it is? And the way you do that is you take a sample, you find the genomic material from the virus, you amplify it, you sequence it, and then you have to kind of figure out how to put that sequence together. Like it's really like a puzzle that the computational scientists are taking care of, where you kind of puzzle back these different regions of the DNA or the RNA to figure out what's the genome of this virus that we have, and then from that you can figure out what it is. That's how we know if it's
Sequencing
a flu
pathogens. Yeah.
outbreak or a measles outbreak or a coronavirus outbreak. And so there's that element of when you don't know what someone has, and it's either spreading or it's causing very severe disease, we use genomics to figure out what they have. And then once we know what it is, we can then do next steps from there, right? We can help figure
Yeah.
out, we can look into developing vaccines or drugs. The other element is when there is an outbreak and you know what it is. Let's say there's a COVID outbreak at your school or a measles outbreak at your school. We want to understand how that outbreak spread. So, for example, for measles, right? Is there a strain that's spreading that it's spreading among people who are vaccinated, and this is actually a strain that's somehow immune to the vaccine, or is it spreading among people who aren't vaccinated? Like what happened with polio, for example. So polio viruses now, there's two types of vaccines for polio.
Okay.
There is one vaccine that's a live attenuated virus. And so what happens is sometimes you'll see with polio that one person gets the vaccine, they actually get a little bit sick with polio, and that's how they build up their immunity. But if that person is then exposed to people who have not been vaccinated, they can actually get polio from this person who was vaccinated. So when we're analyzing a polio outbreak, we want to understand, is this wild polio or is this vaccine-derived polio? And the only way to know that is to look at the genome and look for segments of the genome that will tell you whether it's wild or whether it's vaccine-derived. So there's that element of like to understand how the virus actually spread. We really have to get into the nitty-gritty of this genome and see how it spread. So again, another example of that would be if I want to know with COVID, the common thing was trying to understand, well, let's say 10% of your school gets sick. Did they get sick because they were all partying together and so it was one big event where everyone got sick? Or did they get sick because there were 20 different people who got COVID and then they each spread it to one or two other people? Those are two very different scenarios. The policies that you would implement to prevent that in the future are very different policies, right? Are we going to have a no party policy? Or are we going to say, oh, this really wasn't so avoidable. Maybe we could try wearing masks or something if it's really just coming in from all these different
Yeah.
angles. And so you sequence these people's, their COVID sample. And then from the sequence, you can see how these small mutations evolve. And let's say, like, if you got me sick with COVID, I would expect to see that your COVID genome looks like this, and mine looks very similar, but there's one mutation different. And from that one step, I can figure out, oh, it went from you to me. Whereas if there's 10 steps or 20 steps different, 20 nucleotides that are different between your genome and my genome, we didn't spread it to each other. And so we can
Yes.
kind of start to analyze, right? Like it's a puzzle. It's looking for these clues, these little hints of how did it spread, and then that lets us inform policy. So that's a couple. I know I threw a lot of different viruses out here, a lot of different examples, but just trying to get the sense of like, what do we actually do with this information and how do we leverage it? And then how does it connect back at the end to health policy or to, drug treatments and vaccines?
Definitely, yeah. So, one of your major projects actually involved extracting usable, like, viral genomes from rapid diagnostic tests. Could you explain how that works and why it matters?
Yeah. So I think do you remember early on in the pandemic, like you get a PCR test and they would like
Yes.
swab you. You go to a
Yes.
clinic, they swab you, they keep the swab, and a couple of days later you get your results, right?
Yep.
So for people like us who are trying to find COVID positive samples and sequence them, that was like a gold mine that, oh, all of our patients, the people that we want to get samples from, they're going themselves to a clinic. Everything is centralized. We can just take that leftover material from their test, and now we can run it down our own laboratory pipelines and sequence it. So with the advent of rapid diagnostic tests, which was huge for public health, right? For people to be able to know quickly that they had COVID, right? The difference between knowing 2 or 3 days later, and those 2 or 3 days you might have exposed people, or knowing within 15 minutes if you're positive, that makes a very big difference, especially in more remote areas like Alaska, for example, where maybe it takes a week or more for you to get the results back of your test versus a rapid test was very quick. So rapid tests were huge for just how they kind of changed the paradigm of how we approached testing and knowing and getting our own results. But it made our job much more difficult, where suddenly if there's an outbreak at a school, let's say again as an example, and rather than getting PCR-based testing, the students all do rapid tests. Well, for the school, that's great because now they know in that moment this kid's sick, let's send them home right now. But for us, it's harder because now you have these rapid tests that you just threw out.
Yeah.
How do I get to, I need something to go off of to
They
sequence.
were
I need to have
discarded. Yeah.
some biological material. And so we basically kind of set up a protocol of how would we be able to take this discarded material and still do sequencing from it, right? So, and what's the best component of it to use? So a lot of these tests have a swab. Some of them have a little like tube that you swish the swab into. There's a strip that you usually actually get the result from. So which of these components has the most viral material on it that we can then use to extract and, sequence the genome. And so we found the swabs worked best, which isn't so surprising. It's like the first, it's getting the most content from you.
Mhm.
And then we actually used our protocol when there was an outbreak that was identified through rapid tests. There was an like an elderly facility
Mhm.
in the Boston area where they had an outbreak over the holidays, so around this time. Um, and they tested it by rapid test, which was great for the community because, again, they could quickly tell people their results and get people to quarantine. And then they kept all those rapid tests for us, sent them to us, and then we sequenced them and we were able to say, so the example I gave you before of, is it one big party or is it all these individual
Mhm.
events? We were able to respond here and say, this didn't spread at a holiday party. This spread through individual contact amongst the
Very
residents because there were clearly at least two, if not three or four different introductions of COVID that we could see through the genomes and how similar they were to each other. So yeah, that project was just kind of showing like how do we adapt to a new reality on the ground if we want to continue doing this type of genomic surveillance.
definitely. Yeah. So, also speaking about all of this research that you've done, in 2024 you wrote a paper for the Lancet Microbe, and I read it and I think it really presented some really interesting findings. What were some of the key takeaways from your study?
So the Lancet Microbe, that was this study, right, with the rapid tests.
Yep.
And I would say for me, it's one of the key takeaways. Uh, we didn't show it directly in that paper, but what I would say is if we can sequence from COVID rapid
Yep.
tests, we can also sequence from other rapid tests. So for me, the takeaway is as we kind of move towards a system, as healthcare gets put in the hands of the person where you're doing it in your house, and moves away from the clinic, like we are still able to surveil and understand what's going on. So I think that's going to be relevant for flu moving forward because there's a lot of flu rapid tests now. I think you could also see it being relevant for something like strep, right, a bacterial
Yep.
pathogen that also is tested with like these types of rapid tests that like we are still able to do this type of analysis from
The
this
viability of the
sample type.
sequencing.
Yeah.
Yeah, that makes sense. And how do you see methods like the the RDTs, like the sequencing or the wastewater surveillance you mentioned earlier, shaping like the future of outbreak monitoring, especially in like
Mm.
lower resource regions like that Boston elderly care facility you were talking about earlier?
Yeah, so to focus on wastewater, there's a lot of really promising work being done right now with wastewater.
Mhm.
Um, so I did an internship briefly with the Gates Foundation, which is located in
Oh,
Seattle, and a
yeah.
lot of their focus there is right, where do we invest so that we can build up these capabilities worldwide? And
Mhm.
so they're doing a big push right now on wastewater. And essentially the idea behind wastewater is that if you want to get a sense of what's happening in a community, usually we would go around and ask for lots of samples, right? So maybe you're
Yeah.
standing in a street trying to, ask people, are you willing to take a nose swab so we can just get a sense of what's passing around in the community? What's great about wastewater is that without having to inconvenience anyone, any of the actual people that you're trying to test, you can get this aggregate sample that tells you roughly what's going on in the community, right? So how much COVID is there? How much flu is there? Now, of course, there are limitations because not all of these viruses are shed equally in feces or urine. So you do need it to kind of get into that system for you to see it.
Yes.
But a lot of them, like we really are able to see, like the fact that we saw COVID in wastewater was not really what some people expected. COVID is a respiratory disease, so we expect to
Mhm.
see it in the nose, in the mouth, not necessarily in the digestive tract, coming tract
Yeah.
all the way going out into like your wastewater. So I think with COVID, it was really this pivot of like, oh, we knew that we could use wastewater for polio. Wastewater has been used a lot for polio because polio is an enteric disease, so it causes, right, like you see it
Yeah.
more in that tract. Um, to show that we could do it for respiratory diseases as well was a real game changer.
Mhm.
And now the real push is for individual communities to say, what are the pathogens that we're concerned about? Let's monitor those via wastewater and get a sense of how much is spreading right here. So, right, like, are you concerned about certain bacteria? So, bacterial pathogens in the US, obviously we do have bacterial pathogens, but it's not necessarily as as concerning for us as it is for other countries, right? Salmonella, um, I'm trying to think. Sorry, there's what's typhi? Is it Salmonella typhi? I actually might even look it up right now. Am I saying the right name or am I mixing my bacterial names? Yes, Salmonella typhi um, is like a pathogen that certain African countries will want to look for in their wastewater. We don't have to look for them here. So I think you get to see the system where it's cheaper. You're monitoring more of the population,
So,
and you can look for the pathogens that really matter to you and get a sense of are they on the rise, and then inform policies from there. So kind of quickly going back to COVID, one thing that was really helpful with COVID monitoring through wastewater was getting a sense of how many cases there were, and then hospitals could prepare their emergency departments for an influx of patients, right? So that was a big issue during the pandemic. We just ran out of hospital space for all the people who needed it. And it took a while to kind of get extra wards ready for COVID patients. If you see in wastewater, so wastewater typically you'll start to see a signal before people even are symptomatic. So you could have wastewater, getting a big wastewater COVID signal a week before people have symptoms and actually go test. And then a week after that is when they actually get hospitalized. So maybe you have this 2 week span now where, we saw something worrying in the wastewater. Let's get ready for something. Let's start to prepare to combat this uptick in disease. So yeah, there's a lot of promise there. There's also a lot of work to be done because suddenly you have this huge, complicated data set that like we have to figure out how to interpret and how to actually leverage.
speaking of the data set, you've like contributed to standardizing the white wastewater genomic data, right? Why is
Yes.
standardization so important in this field?
Yeah, so that was some of the work I did through the Gates Foundation. Uh, standardization is essential and it's not pretty, right? Like it's not
Yeah.
the fun work. Everyone wants to be working on the outbreak and showing how you saved people's lives. But if we're all doing different approaches and we're not sharing our data, first off, like it's really important for data to be shared because the more data that's available, the better we can get a sense of what's happening with a different pathogen. Um, But even if you're sharing data, if the way you're generating the data is completely different, you're not explaining to someone else where it came from, how you generated it, what these numbers mean, what methods you used, then the data can be almost useless for someone else who's trying to interpret it and compare it to their own data, their own community. So standardization really is this essential key, like building block
Mm-hmm.
that everything else in public health is built upon. And if it's not there, everything else is going to come crumbling down, right? If we don't understand how to interpret someone else's data and how to trust it, then we can't put all the data pieces together and figure out what's happening, right? And so with COVID
Yeah.
especially, like there was so much communication that had to happen between countries, between labs. We see this also with influenza. Like every year, we have to figure out what's the next strain that we think is going to be the big strain for influenza, so we can predict in advance which vaccine to make, right? Because the vaccines we make a vaccine before the
The
peak of the
strain never
season.
comes out. Yeah.
So you have to every year, right? The flu vaccine, we're just trying to get it as close as possible to what we need. I say we, but I'm not anywhere involved with that type of research. Um, but the establishment, right? Like we're trying to figure out what's the best vaccine that we need for this. Uh, so if people are not sharing their data and not doing it in a standardized way, then you can't use it, and now suddenly there's knowledge. Your knowledge is not complete,
Yes.
right? And if so, it's just kind of this basic, the boring basic building block that everything else rests upon.
Yeah, that definitely makes sense. Yeah. So, every scientific project comes with hurdles, obviously, like you mentioned. What have been like some of the biggest challenges in your research, like technical or otherwise?
Hmm. Hmm. So I think I would say the biggest challenge is is actually always concluding each project, right? So a lot of our projects have a clear public health connection, right? So when
Hmm.
we're working with a university, as I said, when we saw a variant of concern, we immediately communicated that to them. When we were doing the rapid test sequencing, once we realized, oh, these are actually, it wasn't one big outbreak, it was actually multiple small introductions and smaller kind of, groups of people who were spreading COVID amongst themselves, we communicated that back to the public health department. They communicated it to the building. So there's a lot of these kind of immediate results that we know influence public health, and we're able to communicate back. So my current project that I'm working on, we are working on analyzing all sorts of viruses all together. So one single approach that can be applied to any type of virus. And through that, we found a patient who presented at a local hospital in Boston with dengue virus, which is a mosquito-borne disease not commonly found in the US. And we're able to really quickly say, hey, we found dengue. Yes, we know that's concerning because we don't expect it to be in the US, but we were able to also look at its genome and we saw that it was closely associated with genomes from Vietnam. Okay, this, and compared to the clinic, the history of that individual patient, they had a travel history to Vietnam. Okay, no big concern here. This person got this disease while they were traveling. They came back, they were sick. We have not seen it spread past that. There's the immediate public health output. So
Yes.
that part's the easy part. Now, the hard part is at the end, right? We need to be able to communicate our methodology and our approaches and our results to the wider scientific community so that it can be built upon, so that other people can use it, they can see it, they can start to interact with it. And that's the hard part. It's just the hard part is at the very end of the project is figuring out how do I take all these different pieces, all these different things that we did, and what's the most important aspects of that to communicate to everyone else for the future so that 10 years down the line, if it's relevant, someone can go back to this paper and see everything they need to see to replicate what we did or to do something similar or to, compare notes, understand where this sample came from. That I think I would say is the hard part, right? Because like at that point, you're at the end of the project, you're like, okay, I already communicated the public health response, what mattered to public health. I communicated it to them. They know. Okay, but now I still have to tie it all together so that the broader community can also see what we did.
Yes, that is definitely harder, and I can definitely agree with you because you have to find like what part of each puzzle is necessary for your future generations, like the posterity.
Yeah, yeah.
On the
And there's so many
positive
different
side,
things that we do too. Like we
yes.
go down so many different rabbit holes,
Hmm.
just trying to make sure that we found everything that needed that exists in our data set. And so then you've gone down all these different rabbit holes and like, how do I bring it together? Like, how do I take all these pieces and make it one storyline together?
Like you mentioned that 10 months was like a short project time frame for you, like,
10
but
weeks,
still
10 weeks.
10
Yeah.
weeks, yes. 10 weeks is
Yes.
still like a lot of data that you collected though, so
Yeah.
definitely.
Mm-hmm.
On the more positive side, what's been a moment in your research career that so far you found especially rewarding?
Mm, especially rewarding. Uh, I probably have to go back to that very first project. And I think what it was for me, that's where I was working with the university and we really had this like very constant feedback cycles with them. And it was just so rewarding to actually talk to the people who were using our surveillance systems and implementing them and helping people and helping their student body. It was just, it's very easy to get stuck in the science, especially when you're on your computer and you're doing more computational work, where like at a certain point, it just feels like it's just numbers and data that you're working with. you can get really disconnected from the human piece. So I think for me, the most rewarding aspect is whenever I'm really able to connect again to that human piece. And that project, we really had it because we had this really close line of communication where I'm talking to the president of the university from this
Yeah.
university in Colorado. So like, here's what we saw, here's how it's going. Like, you guys did really great here. Like, look, we can show, we can prove that like you kept your students safe and that you tamped down the flow of code on your campus. And we were a part of that. Like, it was just, that was very, very rewarding.
Yeah. Looking ahead, what are your long-term goals? Like, where do you see your work headed in the next 5 to 10 years?
So I'm in an interesting position where, so I just recently finished the PhD in the lab. I'm currently a postdoc in this lab, finishing off the research that I kind of have been
Congratulations.
working on for the past year or so. Yeah, thank you. Um, and I love viruses. I love working in this work, but I've come to the realization that what I most enjoy is working with any type of large data set that is connects somehow to human health. So I can't tell you where I see myself in 5 years from now because I could be in a completely different disease area, doing something completely different. I just want to know that whatever I'm doing, like I can see the connection to human health and that I get to be the one like behind the scenes taking this huge data set and like kind of like, again, like it's a puzzle, right? Like you're taking it and you're kind of turning it from different angles and like trying to figure out like what does this show me? What does it mean? Oh, if I look at it like this,
Yeah.
oh, wow, I see this connection here that maybe people are getting sick, because they have this underlying genetic factor. And okay, if we can address that, we can help them get healthier. Like what is it? How do keep turning this data set that we have until you somehow influence human health? So I could still be in viruses 5 years from now. I could be doing very similar work, or I could be in cancer or asthma or who knows what. I'm really open. That's one thing I've always just been very open to learning about different things. And I kind of like I enjoy getting to be on new projects and getting to like it's fun to not know everything,
Yeah.
right? There's something really exciting when like you're starting something new and you're just so aware of what you don't know, and then you get to like learn about this new disease area or this new, scientific approach and then use it
And
and
like you
help
mentioned,
people,
it's really
right?
exciting to find patterns and like stuff like that. Yeah, definitely.
Yeah, yeah. Like there's that math side of me where I'm like, oh, I'm still at my heart a little math nerd, and I just want everything to like fit into a nice little like plot or, pattern. Yeah. Yeah, I actually liked people complain about organic chemistry because, biologists and many pre-med they all take organic chemistry in college. And I really enjoyed organic chemistry because it was taught very well at my university and it was taught like it was a puzzle. And so you have this puzzle of like how do how do the electrons flow from one part of one part of this compound to another part and how does that change the compound? I'm like, "Oh, I thought this was just memorization. I was not excited, but now that it's a puzzle, I'm very excited about this."
So, many of our listeners are students who are interested in biology, public health, and research, and your work has definitely intersected all three. What advice would you give them?
So, especially for early career scientists, and I would say you're considered early career even when you're in grad school, right? Like, so that might feel like for now, like I might feel like an old person to you, but like this is still early career. Early career scientists, the most important thing is a good mentor. So it is helpful to have a sense of here's what I'm really interested in. I love this type of research. But it doesn't matter if you're doing the research you love if the person who's advising you isn't helping you and isn't helping you grow and isn't involved. So the biggest piece of advice that I would have for high schoolers, undergrads, or graduate students is make sure you are looking at the person who you're kind of signing especially in grad school where like this person's your boss for 5 years, if not more, like you're giving them this huge chunk of your time in your life. Make sure that they're aware of that responsibility that they have and that they're really trying to help guide you. Um, there's yeah, I know a lot of people who like aren't able to continue in the career that they hoped for just because of one bad mentor or one bad boss. You also have to learn how to work with people who are different than you, who are difficult to work with, right? Uh, but I think the biggest piece of advice I would have is try to look past just the cool research and the whistles and bells, and also at the actual person um, who who you are working with. Because at this point, as a high schooler doing research, for the most part, you're going to be advised by someone. As a undergrad, as a graduate student, right, you're advised by someone. So I think just, yeah, look at that piece. The human piece really matters, not just the science.
On a lighter note, when you're working on genomics or infectious disease research, how do you like to unwind?
Oh, that's a good question. So for me, I just try to have kind of a strict cutoff of like, here's my work life and here's the rest of my time. I'm very extroverted, so I just unwind by being with people, and that can be family, that can be friends, but I just need my people time. I like to like I carpool with a friend to work, and that's been like so like for me like I unwind on my way to work and I unwind on my way leaving work because I'm just chatting with a friend in the car. So yeah, that for me, it's just it's people. For a while, I thought that I actually liked, oh, I like tennis or I like this thing. And I kind of realized at a certain point, all of my hobbies that I've had over the years have all had one thing in common, and it was that they involved someone else. It was never, my hobbies have not really, for the most part, been like, oh, I like to sit by myself and like quilt. I do like to read books, and that is by myself, but that's really the only hobby that I do that's solitary.
Yeah, I definitely agree with you. I love reading and I love spending time with other people, like traveling with my family is one of my favorite things to do. So I definitely see how you can also enjoy